This is Dr. Sargisian, and this is second part of Genes and Genetic Diseases presentation. I would like to talk about mutations, and mutation is always a change, and it's not a welcome change, as you know, although sometimes mutation may occur and you will not see any changes in organism or in the behavior or in the appearance or in the functional changes of the organism. So dramatic presentation of the mutation you see in horror movies, obviously not the case many times when mutation occurs in real life situations. So to remember one of the important points here, I would like to say that mutations can be transmitted to offsprings only in gametes. Basically, what I'm trying to say here, when gametes are mutated, or the mutation will be transmitted to the offsprings, that we have a generational transmission, and the children or offsprings will present with a mutation already that occurred in the parents. In somatic cells, the harm can be done to the parent. However, somatic cell mutation will not be transmitted to the offspring. So from the generational point of view, it's important to prevent mutations in gametes in order to produce a healthy new generation. Point mutations is one of the type of mutations that is caused by a single nucleotide base pair change in the DNA. So when we're having a single nucleotide base pair change in DNA, this may have absolutely no consequences because of the redundancy of the DNA, because of the repetition in the DNA. So point mutation may not really have any consequences. Rates of spontaneous mutation may vary, and they vary per gene, and depends on the factors that can also influence the genes at the given point of time. So mutation can be described as anything that alters genetic materials. Chromosome abbreviation or base pair substitution, these are all types of mutations. Substitution of the base pair may result in the change of amino acid sequence, which means that protein will not be produced the way it was designed to be produced. So this can have drastic consequences if we are having change in the protein. However, again, if we are having a silent substitution, this may not result in amino acid change, again because of the redundancy of the genetic code. So when amino acid sequence is changed by a base pair substitution, again, the drastic consequences can occur, because when we're changing the base pair, we are changing the amino acid sequence, we are changing the protein synthesis. Just a couple of words about mutagens. Mutagens are the agents that known to cause or increase the frequency of mutations. Agents influence factor. You can use those words interchangeably, but the main point is something that will cause the DNA to mutate. Radiation is very well known and studied in 20th century cause of mutation. Also chemicals, and by chemicals we have a wide variety of chemicals that can cause mutations. However, some chemicals can be extremely harmful, while others can cause mild or insignificant mutations. The good example of the chemicals are agricultural chemicals, pesticides, herbicides, and fertilizers, but again, the rate of mutations related to these chemicals is very low. A good example of relatively harmless or a weak mutagen is sodium nitrate, and sodium nitrate is a very widely used fertilizer. It doesn't mean that you can put your hand in your can of Miracle Grow and spread it with your hands. Just always use gloves, protection, wash your hands, and protect yourself from even weak mutagen. Silent mutation, again, as I mentioned, is a DNA sequence change that doesn't change the amino acid sequence of the gene. The production of the end protein is what we care about. The way your DNA looks, we really don't care, but we care about changes in protein and with all the consequences that come from it. Missense mutation is a type of mutation that results in a single amino acid change in the translated gene product. Basically, again, remember that protein is nothing but polypeptide chain of amino acids, which are peptides, and if just only one of these amino acids change, then we'll have missense mutation. Can it be drastic? It can, because just changing one amino acid may result in significant change in the protein and call missense mutation or be a consequence of missense mutation. So missense mutation is a type of mutation, as it says here, that results in a single amino acid change in a protein sequence. Okay. Nonsense mutation. This is a type of mutation where messenger RNA stops codon and produced protein will have premature termination of the sequence, or the opposite of that, production of the long protein will happen. So this nonsense mutation also quite disturbing process because it can affect the messenger RNA stop codon, and either your protein is too short or too long. So the main point here is your protein, it's not what we wanted it to be. So it's different protein. Again, not a good thing in genetics. Nonsense mutation is described here in details, and let's talk about premature termination type of nonsense mutation. As you can see here, sentence, the fat cat ate the rat. So it's prematurely terminated, and the fat cat ate, well, the rat is gone, and we don't know what the cat ate, so this is different. You don't want the cat eat something else. And I'm saying this and I'm putting this here so you guys would remember. So this is the sentence turned into a nonsense to help you remember. We had a good sentence here saying the fat cat ate the rat, and now the fat cat ate. What did it eat? Did it go to the pet store and got the cat food? Who knows? So that's the nonsense mutation with premature termination. Another type of nonsense mutation is that when your protein starts getting longer and longer, and when it has to stop, it doesn't stop. So with the same sentence, the fat cat ate the rat, so it turns into another sentence, which is again nonsense. The cat ate the rat and the hat and the mat. So I don't know if your cat eats the hat and the mat, mine doesn't, but you know, this is not typical cat behavior, and you can remember that's it's the nonsense. The sentences are nonsense. So nonsense mutation is related to messenger RNA, and the codon is not activated at the time when it should be. So your sentence, or if we talk in protein language, your protein either too short or too long, but it's not just the right size. Frameshift mutation occurs when DNA is altered and an addition or deletion of base pair took place. So shift is not a good thing in here, because the entire frame, reading frame, so-called reading frame, is transposed and changed. So frame shift, let's say I inserted just one base into DNA molecule, but if you think about it, everything else beyond that one base changed. And every single codon, every single base is not correct. What happens, this definitely won't result into silent mutation. You will see the consequences, because it's just not, although we changed just one base, we added or deleted, but everything had a shift. So everything is different. So frameshift mutation can result in the entire change of the protein appearance and protein amino acid sequence. So let's talk about consequences of mutation. What can happen? Either gain of function or loss of function. So something was dormant, and now it comes up to life, or too much protein product production, overexpression, underexpression, or inappropriate expression. This all can happen because, again, we are talking about protein production, nothing less or more. When we are talking about DNA, as you guys know, it's all what happens. DNA is read and protein produced secondary to DNA composition. So all these gain of function disorders are can be consequences of mutation, and even though these are gain of function disorder, they still are not a positive change. It still can tax the organism very much. Another process that can occur, another consequence of mutation, is loss of function, and it's associated with recessive disorders. So at this time you will lose 50% of protein product, and I won't be telling you why. I want you to think about it, and just, this is an important point, but it's very easy to find. If you don't find the answer, please let me know and I'll help you. Email me or call me. So on the other hand, this is not that bad. Even if you lose 50% of the protein product, it may or may not be adequate for normal function, you know. And there are functions in the body that you really don't need 100% of the ability to function adequately. So, but again, I would like for you to look through this and tell me why this is related to recessive disorders and why, most importantly, loss of 50% of protein product occurs, not 51 or 42, but 50. Chromosomal abnormalities can be quite widespread, and the most common chromosomal abnormality presentation is early miscarriage. In early miscarriage, most of the early miscarriages, they occur secondary to chromosomal abnormalities, and sometimes even this is not detected as a pregnancy. It is so early in the gestation period. But to talk more about this, we need to emphasize the concept of euploid cells. Euploid, or euthyroid, again, prefix in Greek means normal or true, correct, different interpretation. But basically the normal cells have a stable, correct number of chromosomes. That's what we mean by euploid cells. And when we're saying haploid and diploid cells, they still are euploid, that still are normal. So haploid have the half number of chromosomes. So the gametes are the sex cells, or sperm and egg cells, and they are haploid. At the same time, they are euploid. And they have 23 unpaired chromosomes, and I'm sure you all know that, but I have to repeat it just to review the material. Diploid cells have paired chromosomes, and human cells have 46 chromosomes or 23 pairs. Again, diploid cells are euploid forms as well. So in changes when euploid cell has more than double number, it's called polyploid cells. So triploid cells, when the zygote having 3 copies of each chromosome, and 23 times 3 is 69. Tetraploid, when there are 4 copies of each chromosome, it's 92. Just common knowledge in genetics, triploid and tetraploid fetuses don't survive. There is no way this type of fetus with triple or quadruple number of chromosomes will survive. When we talk about lack of normal number of chromosome, we are talking about aneuploid cells. And by saying lack of normal number, I mean this is a contrast to euploid cells. Euploid cells is the multiple of normal 23 chromosomes. So euploid still can be tetraploid, which is abnormal and has no chances to survive, but still it's euploid. Aneuploid means there are not just 23 chromosome multiples. You cannot divide the number of chromosome by 23. So monosomy is when there is only 1 copy of any chromosome, right? Again, remember the concept of pair and pairing. The pair should be there for chromosomes to function normally in somatic cells, of course. So monosomy is when there is only 1 copy of the chromosome. Basically, the other member of the pair is missing. Monosomy is quite commonly fatal. So if there is a chromosome missing from the pair, okay? But if there is extra copy of the chromosome, the infant may survive and live a relatively normal life, and but it's usually the excess chromosomes. Like well-known example is trisomy 21 Down syndrome, and we know nowadays that people with Down syndrome may develop mentally and physically. They can progress in their development, and some of them even go to college and have happy lives. And but to emphasize this notion again, it's better to have more than less, or it's better to have extra than less. If there is extra genetic material, chances of survival increase with genetic abnormality. If there is missing genetic material, chances of survival decrease. And to explain that, you basically don't have enough blueprints to build whatever you are building. If you have too many blueprints, it may be confusing, but you still may be able to build your building. When we talk about chromosomal abnormalities, we are talking about also non-disjunction. And disjunction is, as you know, normal separation. When chromosomes separate during the cell divisions, they go through disjunction. Non-disjunction is non-division, non-separation, and usually it will cause aneuploid cells, aneuploid number. So basically part of the pair or the pair had either disappeared, the member of the pair disappeared, or it went to a different place where it's not supposed to be. So it may cause trisomy or monosomy. So this is the concept of non-disjunctions. I'm sorry. If the homologous chromosomes failing to separate normally during the division, either meiosis or mitosis, that can lead to non-disjunctions. Again, non-disjunction concept is when your genetic material as a chromosome moves to an unusual place and causing monosomy, trisomy. Basically, we don't have euploid cells anymore. We have aneuploid. We don't have multiple of this 23 number, magic number of 23.